over to Dr. Seaton. Sorry.
All right. Well, hey, everybody, thanks for coming. I do have some slides just because it's easier to see things with pictures, but I want it to be informal. So ask questions as you go along. And hopefully, if something's not clear, let me know and I can explain it. Bran, do you want to start the poll to get us started while I'm pulling up my...
Sure. So Dr. Seaton has some polls. I'm going to bring these up. If you have any problems doing the polls, let me know. Here's poll number 1.
So just want to see, Tara, how many people have received the vaccine so
Right.
far?
We have a mixed household. I'll answer for Ricky.
Okay.
Wow. It's a good
I'm
update.
not sure
right?
how that will work, but anyway, you can try.
All right, 100%. So we don't need this talk. You guys know everything there is to know about it, huh?
Okay. Let's go to the second. Do you want the second poll?
Yeah, second poll.
Okay. Here's poll number 2.
If yes, which vaccine did you receive?
Oh, that's interesting. I can't actually respond to the poll.
Yeah, I can't either.
There's no entry for Sputnik.
Yeah, I know. Yeah,
Yeah, I forgot about
not
that
available.
one.
You might
Okay.
have to get in with some politicians. They might be able to help you out. Cool. Most people got Pfizer and Moderna.
Well,
That's
look
great.
at that.
Yeah.
So if I selected, I'd say Moderna. I'm the only one besides one other. Okay.
Wow, that
Good
Cool.
thing, because that Johnson & Johnson is crap from what I've heard.
Well, you know, what they were talking about is just normal reactions, isn't it? But it's too bad we don't have a COVID, a vaccine expert who can talk about that.
Yeah. We can talk about all that later. All right.
Okay.
The last one.
So if yes, are you more than 2 weeks after? Actually, this cut off the text. So the first answer is if you're more than 2 weeks after the last vaccine. The second is you're halfway there. You've been vaccinated, but you're not outside the 2 weeks. And the third is you just got your shot.
Did you go on to Zoom and then press the link to it? Question mark.
Awesome.
Okay, I'm going to end that poll and share the results.
Very cool. All right. So most people have at least 2 weeks after their last dose. That's really awesome. Some
Okay, so
people halfway there. Go ahead.
everybody on this Zoom call can take off their mask.
That's great. That's great.
Okay.
Yeah, for those of you who just got your first dose, yeah, it's at least for me, it was such a relief. So you're almost there. All right, cool. Let me share my screen so I can show the slides. And again, I want it to be informal, so please go ahead and ask questions along the way. So you should be able.
You unshared. Try again. There we go.
There we go. Okay, let me move everybody else out of the way there. Okay, cool. So you should be seeing the full screen, right?
Yeah, it's all set.
Okay, cool. So for those of you, I think I know most everybody, but I work at Duke at the Center for Human Systems Immunology. Hopefully you won't hold that against me, especially during basketball season. But yeah, I really love talking about the vaccines. I love working on them and just helping people understand what they're all about. I know people have had a lot of questions throughout the whole process, which is understandable. So quickly, this is a picture of our lab where we're actually a pretty big group. And, you know, I've been at Duke for 11 years and we've worked a lot on vaccines for infectious diseases. So I just quickly throwing this up here that, you know, people wonder why the COVID vaccines, you know, how they were developed so quickly. It's just, we've learned a lot of lessons from some of these other diseases that we are working on. So for instance, HIV doesn't have a successful vaccine yet, but we've learned a lot about what makes a good vaccine. mRNA vaccines for HIV are in the works and they have been for a long time. So, you know, really the COVID success story is built upon a lot of past work and honestly, a lot of failure and lessons learned. So it's exciting to see that. So yeah, we work on a lot of things, anywhere from HIV, malaria, tuberculosis, all the way up to SARS-CoV-2. And just in case you don't know, that's the virus that causes COVID-19 disease. So we'll talk about the SARS virus kind of throughout. So just to let you know, this up here in the corner is a picture of this virus. So it's kind of it's a virus shaped with a kind of a crown on it, which is why it's called the coronavirus. And so I just kind of tell you guys how it came about. How does it infect us? How does our immune system respond? How were the vaccines developed? And then what about those variants, which I know is kind of in the news a lot, and everybody, you know, is kind of worried about that. This picture is from yesterday, actually. So this shows you from the WHO, the kind of where we stand in terms of coronavirus infections, with darker blue being worse. You can see the United States stands out, Russia also stands out, South Africa and South America. So that's kind of the discouraging piece of the news, but the encouraging piece is that over 600 million vaccine doses have been administered. So that's really exciting, especially because most of them weren't kind of rolled out until, you know, January of this year. So so that's very exciting. The US, of course, likes to be number one in a lot of everything. Unfortunately for us, we are number one in the cases by country, so 30 million infections out of 133 million around the world. And tragically, we're also leading in number of deaths, so 560,000 as of today, out of 2.8 million global deaths. So a vaccine is, you know, kind of our way out. So so I'll go through some of that, but this is kind of just setting the stage for the picture of where we are. So this, you know, disease, it emerged pretty quickly. You know, we are in a pandemic, it spreads very quickly, I think, as we all know. So really, you know, it was only identified January 7th of 2020. And a few days later, China announced the first death. And then March, so 3 months later, it was declared a pandemic. And then about 4 months after it was like the virus was identified, we had 1 million cases worldwide and 100,000 deaths. So you can really, you know, just spread very quickly and really took over. And, you know, as we all know, has caused a lot of devastation. Actually, though, coronavirus, this belongs to a family of viruses that are actually quite common. There are 4 main coronaviruses that cause common cold disease. All of them start out in a natural host, so it's either bats or mice. They might go through an intermediate host like cows or camels or, you know, more rodents. But so it's really kind of surprising because SARS, the one that we're experiencing now, and the one that came a few years ago, really caused severe disease. And I'll go into a little bit about why that happens compared to the others. But we were hoping that, you know, all of us, we have immunity to these common cold viruses, right? Most people don't get super sick from them. We were hoping that would be the case for this virus as well. It hasn't turned out to be the case, actually. So I won't go too much into all the intricate details of the virus, but this is kind of key to set the stage of how the vaccines were developed. This virus has inside of it, this little red squiggly line is RNA. So it's an RNA virus, which is why 2 of the major vaccines are RNA based. And it has this protein on the outside called the spike. It looks like these little thumbtacks or nails. And the key part of that spike is this receptor binding domain. So it's that little red piece on top of the spike that binds to a molecule called ACE2 on the human cells. And that's really what allows the cell to kind of grab the virus in and we can become infected. And this RBD region is the primary target for vaccines and therapeutics. If we can block that binding to a cell, then we can prevent infection. So normally, this is kind of a best case scenario. We inhale a coronavirus like maybe the common cold. It comes into our body and it binds to these receptors on the cell, and the cell kind of engulfs the virus. And the virus, the outer layer, it's kind of oils and lipids, and it kind of falls apart within the cell. And then the mRNA is released, and the mRNA is made into proteins. And so proteins then kind of assemble together and they make a new virus. So our cells become a virus making machine. In normal, if you have normal immunity, what happens is these new viruses are ingested by these kind of like star-shaped amoeba cells in your body. They kind of look out for invaders and they engulf this virus, and that activates your immune system. So there's two main types of cells. There are T cells and B cells. And T cells, they come up after about 1 week. They're kind of that first responder, the major, like, let's get rid of a lot of the threat. They can kill cells in your body that are infected. They recognize the cell's infected, let's get rid of it. There are also B cells, and B cells produce antibodies. They're these little kind of Y-shaped molecules that go around and they grab things that they recognize that are, invaders, and they drag them away, and they prevent the virus from infecting a cell, or it says, hey, this needs to, be incinerated and gone. And it takes about 2 weeks for those B cells to kind of ramp up and become active. And that's why for the vaccines, you have to wait 2 weeks to get maximum immunity. And, ideally, you see something and your body responds to it and you get this long-lived immunity. And hopefully, ideally it happens naturally. But what happens with COVID, actually, the whole process happens again. The virus, you ingest it through your airways, it binds to this ACE2 receptor. Your cells become this virus-producing factory, and it just kind of spreads and takes over, and you get COVID-19 disease. And why does this happen, more than just like a common cold? Well, this picture here shows you all of the areas in green are where you have ACE2 in your body. So lungs, a lot of major organs, your kidneys, female reproductive organs, you have brain, you have your eyes, you have sinus cavities. There's really no place in your body that doesn't have this ACE2 receptor. So when your body is this virus producing machine, it's just kind of taking over and hitting all of your organ systems. And so, this impact for this invader, we all know about the breathing and the lungs. So basically what happens is it just kind of creates havoc in your lungs. All of your immune system starts responding, and your lungs just essentially fill up with like just shredded cells and just mucus and garbage. And that's why people get the pneumonia. ACE2 in your blood vessels and your cells trigger blood clots, they trigger heart attacks, they trigger inflammation. All of that's not good, of course. And then, it can affect it. You can cause effects in your brain, in your eyes. It can affect the nerves in your nose. Your liver goes crazy because your body's trying to fight this off. And then there's a lot of ACE2 in kidneys. So that's kind of another reason why this can be so severe, is you just have a lot of places in your body where this virus can spread out and hide and replicate and just take over. So where does a vaccine come in? So, in natural immunity, you have the infection that leads to the immune response, and that immune response is good, right? We want this immune response to happen, but it doesn't happen fast enough in infection. So we want this immune response to happen without the infection, and that's where the vaccine comes in. You kind of give your body, you prime the pump, it recognizes the invaders, and then it can take care of the infection when it happens. Before I go into any of the vaccine development specifics, does anybody have kind of any questions about the virus itself or effects on the body or anything like that?
So I mute everybody, so feel free to unmute yourself or put a question in chat.
I have a question.
Yeah.
I heard
speak more slowly. I will try to speak more slowly, Sasha.
I heard children have fewer ACE2 receptors, and that's why little people are not strong carriers of COVID. Is that true?
Yeah, it is true. Yeah, they have fewer spaces. They may also, they have less immunity. I mean, they have less, they've seen a lot fewer things, right? So they're not going to go into overdrive either. So, yeah.
Are there questions?
So if it takes 2 weeks for your body to respond, why are we testing people 5 to 7 days after exposure to determine if they've been exposed?
Yeah, great question. When you're tested for COVID, you're tested for the presence of the virus itself. So the levels of virus it takes about 5 to 7 days to show up. The immune response, the antibodies come later. So if you have an antibody test, you would have to wait 2 weeks.
Ah, thank you.
Yeah. Mhm.
Other questions for Dr. Seaton? We have a question from Sue saying, Kelly, do you listen to the TWIV podcast?
I have, yeah, I've listened to that sometimes. That's a good one. That's what This Week in Virology. Yeah, that's a good one. Yep.
Sue loves it.
Yeah, there's a lot of good resources like that out there. So yeah.
Any other questions before she goes on? Okay, take it away.
All right, I will try to talk more slowly. All right, so when we look at COVID-19 vaccines, what do we want? So when we started out with this, what were our main goals? So the first, of course, we want it to be safe and efficacious. No point if it's not going to be safe, and it has to be effective enough to stop the spread of the virus. Clinical trials are very big and complex. And so the initial goal was to look at just severe disease, death, and hospitalization. So if that's all that the vaccine did, it would still be a win. A home run would be that it prevents transmission, so that you don't get it at all. But even if we could just keep the severe disease and death and hospitalizations down, that would be good. The reason why it took longer to understand about transmission is because you have to test people for the virus. And that's really hard to do in such a large trial. They only tested people once they had symptoms. And so that's kind of mainly why we didn't know this right away. In terms of inducing immunity, we want high levels of these antibodies so they neutralize the virus and the threat, or they can block it binding to ACE2. So here in this little picture, the antibodies are surrounding the virus, and it can't even get to the cells to infect. We do want some of those T cells. They can kill the infected cells. So if some does get through, it can kill those cells, and then they can't keep spreading it. And we wanted long-lived protection. So we want something ideally like hepatitis B, which would be a lifelong protection. Seasonal influenza is kind of annoying. You have to get a shot every year, and tetanus, is once every 10 years. So hopefully between 10 years and hepatitis B is kind of what we would be good. And then we also needed them to be scalable and rapid manufacturing, right? We're in the middle of a pandemic. If it takes a long time to make it, or it doesn't meet high quality standards, then we couldn't use it. We wanted to minimize the cold chain requirements. So, when they first came out, you might have heard, right, that, they need to be stored at ultra cold. And they need to be, somewhat stable. And then we want 1 or 2 doses, right? No one wants to get more than 2 doses of a vaccine. At least I don't. Problems with, follow-up, remembering to get your shot, or, just not wanting to do it. So there are actually multiple vaccine types. I'm glad somebody mentioned Sputnik. There's a lot of different ones, and the major 3 that we have in common. This is a lot on this slide, just to let you know there's a lot of different types of vaccines. We're going to focus on 3 that are going to be available in the United States and Europe. The first, which you haven't heard about yet, is a little farther behind, is Novavax. There's been a little bit of news about it. This is basically giving the spike itself. They have a protein. They put that's the vaccine. You give the body that spike on the outside. It's just a piece of the virus. It can't give you COVID, and your body recognizes that. And then if you see the virus, then you have immunity. Janssen, which is J&J, and AstraZeneca, they're slightly different. These are replication incompetent vector vaccines. So what they do is they put a piece of the coronavirus genetic material into some other virus. So usually this is an adenovirus. Adenoviruses are cold viruses that are inactive, so they can't give you a cold, but they're very efficient at delivering genetic material. So your body can then recognize the coronavirus piece that's inside the vaccine. And then finally, the ones that have gotten a lot of press are RNA vaccines. So basically you put the RNA that's makes up that spike into a piece of lipid or oil, and then you deliver that, and then your body makes the vaccine or makes the spike protein, and that's what Moderna and Pfizer do. So I'll go through each of those three, and these are likely to be what's going to be available in the US as time goes on. So again, for mRNA vaccines, they train the system for a real infection. So you have this RNA, it gets engulfed by these star-shaped amoeba-like cells. They then see this mRNA, it's like a message. It tells the message, it says, make this protein. This cell makes the protein, and then your body, the T cells and B cells are activated, and you have in your system the antibodies and the memory cells. So when you see the real infection, you're ready to go. You don't have to ramp up. And there's a couple pros for these. So for Pfizer and Moderna, they're very rapid to make, and you can scale them up very, very quickly. I think it was about 30 to 60 days after the sequence was discovered that they put this in, they started manufacturing it. You can easily adapt it. So it's very simple. So you can change the sequence very quickly as the new mutations come up. And there's minimal ingredients. This is one of the best things about this vaccine is there's mRNA, there's salt, there's lipids and fats. All three of these types of ingredients, all of them in the vaccine are found naturally in your body. And then there's a polyethylene glycol, which is a kind of a solubilizer to keep everything in together, when it's injected. And that's very commonly found in cosmetics, in lotions, in food products. So the likelihood of allergies to any of these are very low. The downsides, cold chain requirements that you need to be kept very, very cold. And then there was a lot of hesitation because these are the first vaccines brought to commercial licensure for mRNA vaccines. The technology's been around for a while. There just wasn't anything that was so urgent to get it out. It's been developed for Zika, Ebola, for some others, looking at it for flu and for HIV, but nothing's actually been licensed yet. So I think there was a lot of hesitation around it because of that. J&J and AstraZeneca, they have a lot of pros too. So this is where you have COVID genetic material inside another virus to deliver it. The pros for this, it's been developed before. It's easily scalable. It grows up pretty easily, and we know how to dose it, right? Because it's been around for a while. Then downsides to it is that could be some pre-existing immunity. So adenoviruses are very common. People might already have antibodies to the delivery system for this vaccine. And so, so that's kind of one of the downsides of this. But they picked adenoviruses that are different than the ones that are kind of commonly circulating, so that would hopefully be minimized. And then Novavax. So this is a protein-based vaccine where you're just injecting the protein into people. And pros for this is most of the vaccines we currently have licensed use this technology. And we know what carrier molecules can improve the responses, make them last longer. The cons for this is very time consuming to manufacture. And there's a lot of vaccine components that have to be quality checked along the way. So the more ingredients, the more you have to quality check, and they're more complicated to manufacture. So they're very effective. We know how they work. It's just really that manufacturing piece that is a holdup to getting this out quickly. So how did we do? The vaccines are safe and efficacious. I mean, that's been very, very overwhelmingly shown. I mean, the efficacy is amazing, and I'll go through that a little bit. The primary was really out of the park, preventing severe disease, death, and hospitalization. The secondary, preventing transmission, is very promising, and I'll show that as well. That, if you get the vaccine, you're protected from getting COVID or SARS-CoV-2 infection at all. How did it do in terms of getting immunity? We had very high levels of these antibody responses. So, I've heard a lot of people, I don't need a vaccine, I've had COVID, I've got antibodies, I'm good. Well, the vaccine gives you 10 times higher levels than you would if you got infected with COVID. So, your immunity is higher and it lasts longer. Your immunity is also probably better quality. We're still looking at that. It gives us the T cells that we want. And the long-lived protection is also very promising. We've got new data showing that the how long it lasts is just as effective 6 months after vaccination. I think it's going to be even longer. We see really no drop in immunity after 6 months. So all of that's like super exciting. This kind of graph is very exciting for scientists to see. I'll walk you through it, see if, in case you're not familiar with these. The blue line in this case is the placebo for Pfizer. And this is essentially over time, how many infections do you get? And you see over time, this blue line just kind of rises straight up and kind of almost turns exponential. So this is if you have nothing, no vaccine. What you see on the bottom here is this kind of right-hand turn. So before that immunity comes in for the first 2 weeks, it starts to rise and then it just flattens out. So you're not getting any infections over time if you have the vaccine, but this gap in between is a really good sign the vaccine is working. Same with Moderna. The purple line, you do nothing, it kind of just rises over time and you just keep going up. You get the vaccine really, really flat. After the first 2 weeks, you don't see the rate of infection going up. And you see that here, 15,000 people with a vaccine, 15,000 people with a placebo, 11 infections with a vaccine, 185 with a placebo. These are really, really effective. This just came out from the CDC this past week. They prevent infections in real world conditions. So they looked at 4,000 healthcare personnel, first responders, essential workers. These are people who are exposed to the virus and they were tested weekly. And then those who are fully vaccinated were 90% less likely to get infected. So that's pretty amazing. Your flu shot any year to year might be 60% effective. We've got an HIV vaccine, it was only 30% effective, so we have to tweak that more. This is a home run out of the gate. So how did it turn out in terms of like the manufacturing and logistics? So we've got it scalable, we have it rapidly manufactured, it meets the quality standards, all of that is good. The cold chain requirements, depending on the vaccine, that is still kind of a burden, but it's manageable, at least in the US. And then it's a 1 to 2 dose regimen, so that was great. Does anybody have any questions about kind of the effectiveness? The next piece is a little bit about how did it get manufactured so quickly and developed.
So Kelly, we have a question from the chat from
Yep.
Dr. Schick. How do you define better quality immunity?
Yeah, better quality immunity is a stronger response. It could recognize more, it could be broader, so it recognizes more variants. And it also, when antibodies bind, they have this thing called affinity or avidity. So it's how tightly that little Y shape, the hands on that Y-shaped molecule grab the virus. In early infection, they kind of recognize it and they grab it. It's kind of, sometimes it's a little bit like those little robots that you go around and trying to pick up that toy, right? You could get one that grabs real well, or most of the time it could fall right out. So better quality is you kind of move away from that, trying to grab the toy out of the vending machine to something that you recognizes it tightly.
Other questions?
I have a question.
Yeah.
Sasha has a question.
Sasha, go ahead.
If you got one vaccine, is there any benefit of getting a second vaccine that has a different mechanism?
So you're saying mixing and matching, or?
Well, say you got Pfizer, but then you got Novavax or something. Is it any more beneficial to have them both?
No. For the 2 doses, you need to have the same manufacturer or the same type of vaccine if you can't get the same manufacturer. For booster shots, we don't know yet. I don't know if we're going to need boosters. If enough people get vaccinated, the virus will actually die out and we won't need a booster. That's what I'm really hoping for. But I'm sure people will look at that and see, what type of booster could you get. But no, I don't think people need to go get multiple vaccines.
Judith, go ahead.
Oh, I've been really curious about why they're making such a big deal about kids being different. Are they worried about it being more dangerous for children or less effective? Why do they need separate trials for children?
Yeah, so they need to get the dosing right, actually.
What?
So kids don't always need the same dose that adults do, partially because of size, partially because their immune systems are not, they're not the same. They may not have matured quite to the same level. Yeah, so that's
So it's
why.
more a matter of efficiency, but it's not dangerous, is it?
No, I don't think it's, yeah, so far the Pfizer vaccine has not been, it's very safe in kids as well. Yeah, it's not dangerous. It's more about how, what dose do we give them and how do they respond to that? Yeah.
Other questions? Just turn your mic and belt it out.
So where they did have children, they've gone down to age 12. Have they had to adjust the dose? I mean, a 12-year-old is smaller than an adult, right?
Yeah, I have to look at it. I don't know for sure, but I think they did drop the dose a little bit. Yeah.
You think what?
I think they have dropped the dose, but I haven't seen the number about how much, I think they give the children smaller doses and they get the same types of responses as adults. Yeah. But I have to look that up. I don't know the number.
Anybody else? Dr. Longley,
I
your lips are moving.
have a question.
Go ahead,
Yeah.
Flo.
Why is it that some people develop a strong reaction after the second shot and others have nothing?
Yeah, it's just, I think it's how much their body, so the mRNA is a message, right? And your body's producing protein. I think it just depends on how efficiently that person produces the protein. Some people might produce more protein and then the body reacts stronger and some people may not. I think as people age, the immune system is not quite as strong. And so older people tend to have fewer side effects for that reason. Their immune system is not responding quite as strongly. It still responds very well to the vaccine, but not quite as strongly.
So the sicker I get, the better it is for me.
Yeah.
So it's not related to how seriously you would get the disease. I made the assumption it would correlate with that.
No, it doesn't. Yeah, no.
Golly.
Yeah.
I was wondering if you think that people who have allergies are more likely to have secondary things, reactions to the virus, to the vaccines.
Yeah, so I'm not a medical doctor, so I have to have that disclaimer. I can't really speak to any particular allergies, but especially for the mRNA vaccines, like I mentioned, the components of the vaccine are mRNA, which everyone has in their body, salt, fat, and yeah, so there's nothing there that should cause any type of allergic reaction. The PEG would have to be a pretty specific allergy because we encounter that pretty much in our everyday lives. And so, if you use lotions or you use, soaps or anything like that, you're exposed to that. And if you don't have a reaction to that component, then yeah. So, but seasonal allergies, like I think bee stings and things like that don't seem to be a problem for this. If you are allergic to other vaccines, they do recommend to ask your doctor and see if this one would be right. Yeah.
Is there any expectation Novavax is going to come along as any great usage?
Yeah, I think it will get, it's working its way through the approval process. I think the US is pretty far ahead of other countries, so we may not need Novavax, but yeah, I think it should be available for use, yeah.
Other questions for Dr. Seaton before we go on?
You didn't mention the Johnson one. Which one? What is that like?
Yeah, Johnson & Johnson, yeah. So that has the adenovirus vector. So that is the COVID genes inside of this other virus to deliver it. Yeah, so it has less effectiveness than the others, but it still has really good effectiveness. It's only one shot as well. It does better against the strains in the US and the UK than it does the South African strain. So, you know, the advice, and I agree with it, is that if you are offered a vaccine, take it. There's specific scenarios, I think maybe traveling a lot, like I travel to South Africa quite a bit. I wouldn't want to get J&J. I got Pfizer instead, that was what was offered, but for most people, it's just as good as some of the others.
Okay, thank you. Let's go on.
Yep. So, you know, I think as this has come out, a lot of people wonder, you know, how did this get made so quickly? It just seemed like it came out of nowhere, right? No, vaccines take years and years, and they do. So this on the top is traditional development. You have to have design, you have studies to test, you know, how does the vaccine work, first in small animals, and then it moves forward, and then, you know, they submit a lot of paperwork. An IND is a new drug application that goes to the FDA. The FDA takes some time to review it. You have a phase 1 trial, which lasts 1 to 2 years, that says this is safe. It takes that long mostly because of recruiting people, right? I mean, how many people around here are looking for trials to sign up for an experimental vaccine, right? For something like Zika, which you're not going to be affected by. No, you know, most people, it takes a while to recruit. And then they do some analysis, they do the next stage. Phase 3 is kind of that big efficacy. Does it actually work in a real world? There's some more paperwork that takes a while. And only then do they start producing the vaccine, right? Because no one wants to produce a vaccine if you don't know that it's safe, if you don't know that the FDA is going to approve it. So this whole process could take, you know, 15 years or longer. The way that this could be developed so quickly is that a lot of this initial design and testing had already been done for the SARS, the first SARS, or for MERS. The FDA and everybody else dropped everything to review this. So, you know, paperwork that took months, took weeks, because that's all they were focused on. And then they compressed the clinical trials. So they did make the phase 1, the safety, you know, they took the time to make sure it was safe, and then they started the next trial once they knew it was safe. And in clinical trials, they review data every month. So for safety, so if they see that it's not safe, they don't finish that trial. So it's regularly reviewed. And one of the biggest things, and I'll show that on the next slide, is that the US government assumed responsibility to produce these vaccines before they knew they would actually work. So if they turned out to not be effective, we would waste millions and millions of doses. But if they were effective, they'd be ready to go. So that was kind of one of the major reasons this could be done. Everything was being done simultaneously rather than sequentially. And this review by the FDA took, you know, a shorter time. Again, they dropped everything to focus on just this, and they reviewed it on a rolling basis rather than waiting till things came in at the end. And this was really done by Operation Warp Speed. So this was announced in May of 2020. And again, they took 15 months. This brown bar is manufacturing. They spread out the manufacturing all the way through clinical testing. Now, again, you know, the US government, taxpayers were on the hook, right? If this failed, we would waste millions of doses, millions of dollars. But if it succeeded, which it did, we'd be ready to go. And I think that's one of the key things why it was done very quickly. No corners were cut in terms of safety, the number of people being tested. People had incentive to sign up for the trials, right? We had some Solterrans sign up for trial. And, you know, and so everybody was motivated to get this done quickly. And, you know, mRNA, again, this is an advantage for mRNA. It took 63 days from the sequence to first in human. And then it was very quickly moved through all of the testing, including, you know, over 30,000 people in the clinical trials. And I like this quote by Dr. Kizzmekia Corbett. You know, she says, producing these proteins takes more than a year, but manufacturing this mRNA can be achieved in a matter of weeks. So they have advantages in manufacturing speed, and they're very good at generating immune responses. And I love putting this up because she's actually a local. She grew up in Hillsborough and went to UNC for her doctorate. So it's very exciting. We have someone local who really just championed this and got this through. This is the Moderna mRNA vaccine. So Hillsborough in January proclaimed January 12th is Dr. Kismekia Kizzy Corbett Day. And I've met her and talked to her. She's a very lovely person. So I think that's an exciting local connection to fighting this pandemic. All right, so I'm almost done. Just talking a little bit about the variants and vaccine efficacy. We've seen a lot coming out recently. You hear about all these variants, right? There's three main ones of concern: the UK variant, South Africa variant, there's a Brazil variant, and a California variant, actually, that's fairly new. All of these are more transmissible, so the infection spreads faster. UK variant is likely more deadly as well, and the others are possibly more deadly. We still don't know quite yet. But again, they spread faster. These are not actually the first variants. The first one occurred in spring of 2020, and that's really what caused the virus to just take off worldwide. And so variants have been around. They've gotten a lot more press in the last, since last fall, because they are adding more transmissibility on top of an already transmissible virus. So the good news here, the spring 2020 variant, all vaccines are effective because they were developed with that in mind. The UK variant is unlikely to be a problem. We see a small drop in immunity from a recent Duke study, but it's unlikely to be a major problem for the vaccines. The South Africa variant is more problematic. So AstraZeneca was going to be rolled out in South Africa. They stopped rolling it out there because it has no efficacy against that variant. There's some drop in immunity for Pfizer, Moderna, Johnson & Johnson, but they're still very effective. And they're extremely effective at preventing severe disease and hospitalization. The Brazil variant is still to be determined. Early studies are promising, but we just don't have enough data on that yet. And the California variant, which is good, the good news is that's also unlikely to be a problem. So we see a small drop in immunity. So I think the bottom line is really encourage everyone unless they have a health reason to not get a vaccine, to get a vaccine. It's going to stop the spread of the variants. It's safe and effective. Yeah. And then very quickly, what's our ongoing research? So our lab in particular is studying the responses in natural infection, and we can learn from that. So there are some people that are asymptomatic and some people that get severely ill. And so what we want to know is what naturally causes one person to get sick and one person to not get as sick. And if we understand that immune response, we can kind of tailor make the vaccine to go that direction. We want to look at how long immunity lasts. So far, we've seen no drop in effectiveness for at least 6 months. And then we do want to look at people like who have autoimmune disorders, pregnant people, really just to see how do these vaccines work in those populations, and can we make that more effective? I think it's an enormous challenge to make a vaccine and do all this research. And really, one of the great things is the US government and Bill and Melinda Gates Foundation and all of these networks have come together to really just throw everything at this virus and to stop the pandemic. And a lot of these are people who already work in the vaccine field for other diseases. So, I think the US as a whole has the best scientists working on it through US government and again, private foundation partnerships. And they really stepped up to the plate to do all this work. So yeah, encourage everyone to get the vaccine, and I'll answer any more questions if anybody has them.
So Kelly, I have a question.
Yeah.
What is your specific work?
So my specific work is looking at antibody responses. So the quantity and quality of antibodies that are generated, both when someone's infected and also when they're vaccinated, and then comparing those as well.
Got it.
Yep.
Anybody else?
I have a question.
Yep.
I thought I understood that some people who did not get symptoms for getting the virus were still getting internal damage happening. Mm-hmm. Does that still happen with the vaccine and being exposed?
No, so the vaccines don't actually do any damage. They just train your body without getting the damage because they can't bind to that. They can't infect your cells, right? The infection is what causes the damage. Part of what makes someone sicker is the dose of the virus. So if you get really like a big blast of virus, you're going to be sicker. But that's not the only thing, but that's part of it. That's why they want people to wear masks, right? You can reduce the amount that you get.
Next question.
I have a question.
Yep.
Does your research or your group inform you about therapeutics? It's something that might work as a therapeutic.
Yeah, so antibodies can also be used as therapeutics. So they've taken really good quality antibodies from people who've been infected, and they've kind of purified that out. And then they can infuse that in, they can grow that in the lab and then infuse it to other people, and those work very well. That's kind of like the monoclonal antibody therapy. I think President Trump got that when he was in the hospital. I don't know as much about kind of the drugs side of it, like the antivirals. There are some antivirals that work well too, but I'm not as familiar with that part.
Okay, thanks.
I see Brenna's unmiked. Brenna, go ahead. Or Preston, sorry.
Sure. Yeah, I was going to ask. I've heard there was like a sort of unprecedented sharing of, as you sort of mentioned in the end, and yeah, I'm just wondering of resources and knowledge that, like, do you think it was different from before, even from a university perspective, and like and sharing with these private companies? And I'm always kind of curious how the private companies get to, well, they get patent rights, and then, but do you think like a lot of the research was done, was the R&D done by these companies, or I guess in universities mostly, or these other organizations?
That's a great question. So the kind of the technology existed pre-COVID, right? They're just kind of tailoring it for COVID. So Operation Warp Speed, all the companies that participated in that got money for development had to agree to kind of play together, right? And some of them are even manufacturing the other person's vaccine, the other company's vaccines, right? In terms of coming together, it was unprecedented. Like, we had someone say, the world should tackle one disease at a time. Like, everyone work on one disease, solve that, and then move to the next one, right? Because we got it done so quickly, and it's been exciting. It has. So for Operation Warp Speed, Pfizer did not join Operation Warp Speed for development. They kind of wanted to go their own way and faster. They put their own money into it. They did take money to distribute it. But yeah, the other companies had to agree. And we worked quite a bit with Moderna. Moderna in particular is very open. They're like, we just want to help solve this problem and get it out there. And, they're protecting their patents, but they're not overly secretive about anything. So that's been really good.
Maybe the next disease you can tackle is republicanism. So, so I see Bennett is unmiked. Bennett, go ahead.
Kelly, what was the focus of Duke's work in all this?
So Duke has done a lot, actually, not my group, but there's David Montefiore. He's often in the news. His lab tested all of these vaccine trials for the effectiveness. So they've got thousands and thousands of samples they test. They're working 24/7. Duke also has done a lot in the monoclonal antibody therapy world, and Duke is also developing a vaccine that will protect from common cold and COVID, and a broad coronavirus vaccine. So yeah, so Duke has done quite a bit, actually. Yeah.
I have a messy question from chat from Barbara. Kelly, thank you. Great presentation. At what point can families who have been vaccinated feel comfortable allowing vaccinated visitors in their home?
Yeah, so 2 weeks after everybody's got their last dose, that's no problem. The vaccines are pretty effective after the first dose. So I wouldn't panic if someone comes to your house. If someone's vaccinated and you're halfway there, I wouldn't panic. If they came unmasked. But the official recommendation is 2 weeks after the last dose. Then it's completely fine.
If everybody's 2 weeks after, then we can all kiss and hug and have a grand old time.
Yeah.
Okay. Don Horstman's unmuted.
What?
Did you have a question, Don?
No.
Okay. Other questions?
Yeah, I have a question. Oh, sorry, I don't want to jump in front of anyone else. Can you talk a little bit about the blood clot issue? I don't know if that's not probably way off your area of expertise, but I'm just curious to see like how much of an issue you think it is versus like, because I hear some statisticians saying like, oh, the probability of this happening is low and lower than you'd expect in the natural public, regardless of COVID. So
you're talking about it for the vaccine, the AstraZeneca vaccine?
Yeah,
that causes
exactly.
blood
Yeah.
clots? Yeah. So the European version of the FDA is likely going to put a black box kind of warning on it that like your risk is very small, but it does happen. Yeah, it's a very rare disorder where you get blood clotting and a drop in platelets. It's still very rare, but it's associated enough with a vaccine, they're going to put that warning out. AstraZeneca is not in the US yet. So yeah, it tends to happen in younger people. So I think the European group is going to say older people can get it safely and younger people should think about it. Judith looks like she has a question.
Well, I heard after the fact that if you were on regular doses of aspirin, you should start taking it when you get the vaccine. So I wondered if that was what was causing the blood clots.
I don't know what's causing the blood clots, but generally, yeah, you should avoid taking anti-inflammatories before getting the vaccine because that can help. Your immune response might not be as strong. Although if you are on it regularly, I believe you could still take it, but yeah. I don't know particularly related to the blood clots.
Rebecca, you had a question?
Yeah, so along the lines of, I guess, determining effectiveness among different population groups or male, female, or whatnot. I remember back when the virus first was wreaking havoc, I remember reading something around that blood type might matter. Can you speak to, do we know like if that is in fact a factor in the severity of the disease, and along those lines, do we know if blood type matters in terms of effectiveness in a vaccinated adult or
yeah, so in terms of infection, yeah, I heard that a lot in the beginning of the pandemic. I haven't heard anything about it since then, so I don't know if my feeling is it was a finding in a small number of people to begin with, and then it turned out to not be a case in the larger population. I don't know that for sure though, but I haven't heard anything about blood type recently. Blood type shouldn't matter in vaccination and your effect. Yeah, blood type would not be relevant to vaccination.
Okay, cool.
If you have a question for Dr. Seaton, just unmute your mic and I can see you.
Yep, Judith.
I heard a lot initially, not so much recently, that the severity of disease was actually dependent on the response of the immune system. Is that true, or did that get disproved?
Yeah, so some people get severe disease because their immune system just goes haywire, right? Like they go into like this shock, and that's still true. Why their immune system goes haywire is not well known.
I also heard that if you had certain Neanderthal genes, that might have heard that.
Yeah, I don't know about the genetics. Yeah.
Going back to the question of aspirin, aspirin has blood thinning qualities. So if anything, you would think that would be somewhat preventive of clotting.
You'd think so, yeah.
No, my point was that people were told to stop taking their aspirin.
Yeah, I don't know in terms of the people with blood clots. So there's 58 people with blood clots after they had the AstraZeneca vaccine. Not all, I think 58 that were associated with the vaccine, and 19 people, I think it was 19 people died. But again, that's 58 people out of millions and millions of doses. So it's still pretty rare, and I don't know if they know yet why those people had that disorder.
Other questions? Going once, going twice. This has been one of the greatest talks we've had. I really appreciate it.
Thanks.
And we'll have to have you come back when you work on another disease.
Oh, well, yeah, I have lots of diseases I'm working on, so
Yeah.
Thank you all for your attention. You guys had great questions. So if you see me walking around, I'm happy to answer anything else you think of. So
Thank you, Kelly.
Thank you.
Thank you so much.
This was great.
Bye all.
This is awesome.
Very informative.
thank you.
Thanks, Kelly.
Thanks, everybody.
Bye.
All right. Kelly, that was great.
Good.
I'll
Thank
have
you
this
very
up
much.
for everybody else in 24 hours. It'll be up on our YouTube channel.
Okay.
Thank you, Kelly.
Thanks. No problem. Yeah.
Bye-bye.
Bye, y'all.
Thanks.